Welcome back to my channel, friends. In today's video, I am going to explain on fertilization. Okay, fertilization in Cycas plant. How does fertilization occur? So here, now in fertilization, that means at the time of fertilization, what happened actually? The nucellar tissue, you can see here, this is the nucellus. Okay, the nucellar tissue between the pollen chamber. Here there is a pollen chamber, and this one is the archegonial chamber. So you can see here clearly that the pollen chamber and the archegonial chamber here, the nucellus in between these two chamber, it becomes disorganized. Okay, and simultaneously we can see that the ventral canal nucleus will also disintegrate. So what happened in the first phase of fertilization is that the nucellar tissue in between the pollen chamber and the archegonial chamber disorganized, and also the ventral canal nucleus also disintegrates. Now we can see here that the pollen tube they reach the pollen tube reaches the archegonial chamber. You can see here, this is the archegonial chamber. Now the pollen tube, it reaches the archegonial chamber, and the tip of the pollen tube, it ruptures. Okay, it ruptures, releasing the two male gametes and also the fluid contents. So you can see here, this is the archegonial chamber, this is the pollen chamber. The nucellus gets disorganized and the pollen tube reaches the archegonia, where it releases the fluid content as well as the two sperm cells, that is the male gamete, into the archegonial chamber. Now due to this, the archegonial chamber, it becomes moist, and the sperm move freely in it with the help of cilia. We know that the sperm cells, they are having cilia, so they can move freely in the archegonial chamber with the help of the cilia. Now the sperm, after they swim in the archegonial chamber within the pollen tube, they reaches the neck. Okay, they reaches the neck of the archegonia. Here you can see these are the two sperm cells which already reach the neck of the archegonia, and here they will force, okay, they force their entry into the egg. Now only a single sperm enters violently in each archegonium through the neck. Okay, through the neck cells, a single sperm will enter, and the cytoplasmic membrane and cilia of the sperm, they are slipped off, and its naked protoplasm is drawn towards the egg by chemotaxis. Now you can see here, okay, this the sperm cell, it already reaches the egg nucleus, and the naked protoplasm is drawn towards the egg by chemotaxis. Now the sperm nucleus, it finally fuses with the egg nucleus to form a zygote or the oospore. Here we can see, when it reaches the egg nucleus, they will fuse together and they will form what we call a zygote or the oospore. Okay, so which is diploid, the oospore or zygote which is diploid. Now because we know that the egg nucleus is haploid, as well as the antherozoid nucleus is also haploid, So when they fuse together, they will give rise to a diploid zygote. Now the fertilization in Cycas, it takes place with the help of the motile sperm. Okay, the motile sperm, we know that the sperm, they are motile and can move with the help of the cilia. This process is known as zoodiogamy, and it is also accompanied by the formation of the pollen tube. Okay, it is also accompanied by the formation of the pollen tube in a phenomenon known as siphonogamy. Okay, so zoodiogamy is a type of plant reproduction in which the male gametes or the antherozoids, they swim in a path of water towards the female gamete. So that process is termed as zoodiogamy. And siphonogamy is a condition in plants in which the pollen tubes are developed for the transfer of the male gametes to the egg cell. Here in this case, we know that the pollen tube, they serve the function of transferring the male gametes, the antherozoids or the sperms towards the egg cell. Now, here the rest of the male nuclei which are present, they will degenerate, and this is termed as polyspermy. Okay, so polyspermy, it describes an egg which has been fertilized by more than one sperm. So next we will take a look at the development of the embryo after the zygote has formed. We will take a look at the development of the embryo. Okay, now the development of the embryo after fertilization, the zygote enlarges in size. We can see here, this is the zygote in figure A. Now in figure B, we can see that the zygote enlarges in size and its nucleus, it divides by free nuclear divisions. Okay, by free nuclear divisions into numerous free nuclei. 64 to 256 nuclei. Okay, now these nuclei, we can also see here that they are distributed in the cytoplasm. They are distributed in the cytoplasm. So what happened next? Simultaneously, a vacuole, you can see here a big vacuole appears in the center, and thus what happened? All the nuclei, they get pushed to the periphery. Okay, here the nuclei, they are distributed in the cytoplasm. Due to the formation of a large central vacuole, these nuclei, they get pushed to the periphery. Now there, what happened actually, you can see here that just remember this, that more nuclei are present on the lower part. Okay, more nuclei are always present at the lower part, that is the chalazal end, and also the micropylar end. Now in Cycas, the embryo is formed. This embryo, it is formed from a cellular part of this chalazal end. Okay, from this chalazal end, the embryo will form. Hence, this structure is termed as the proembryo, but not the embryo. Okay, so the proembryo, it is the series of cells that are formed after fertilization of the ovule. Okay, after fertilization within the ovule of a plant. Now this is termed as the proembryo. So after the formation of the proembryo, a wall will start to form. Okay, whorl formation, it will begin at the base of this pro-embryo, at the base, okay, which gradually extends up to the periphery until the entire structure will become cellular. Now the basal of the pro-embryo are smaller and more numerous. Okay, this base part of the pro-embryo, they are small and numerous with a dense cytoplasm. And it is from this basal part that the embryo proper is formed. Now the upper cells of this embryonic region, as you can see in figure F, the upper cell of this embryonic region, they will begin to elongate vigorously, forming a long spirally coiled structure called a suspensor. Okay, this will form a suspensor. You can see here in figure E where they start to elongate, and here they are much more elongate in figure F, and this will form a suspensor. Now a suspensor, what is the function of the suspensor? It will actually serve to push the developing embryo into the endosperm. Okay, so here some of the nuclei in the upper region, they remain without cell wall. Okay, you can see here there is no cell wall in the upper region. Now the pro-embryo will soon get differentiated into 3 zones. Okay, the upper one is called the haustorial zone. Okay, at the micropylar end, this part will form the haustorial zone, and the middle is the suspensor zone, whereas the basal part at the chalazal end will form the basal embryonal zone. So you see it has already differentiated into 3 zone: the embryo, the suspensor, and the haustorium. Like I said, the function of the the suspensor, it actually serves to push the developing embryo into the endosperm, whereas this haustorium, it will help to absorb the nutrients and water. Now the haustorial zone absorbs the nutrition for the developing embryo. Okay, so the embryo with 2 prominent cotyledons. Okay, let me show you. Now you can see here, this is the embryo, the major embryo, where you can see 2 prominent cotyledons which develop from the tip cells through a series of division. Okay, it will lead to the formation of the cotyledons. Now the cotyledons is an embryonic leaf in seed bearing plants. So here, due to the development of a long suspensor, the embryo is pushed out of the zygote membrane deep into the endosperm. It is due to this suspensor, it is pushed out of the endosperm. It is pushed out of the zygote into the endosperm, from which the embryo will derive its nutrition. Now the embryo within the ovule, it gradually enlarge greatly. Okay, it will gradually enlarge greatly. Now a plumule, later on a plumule, that is a rudimentary shoot or stem of an embryo plant, it will start to form. Okay, it is then differentiated in between these cotyledonary depression. We can see here you can see here this plumule. Okay, first we see the formation of the cotyledon, and later on a plumule is differentiated between the cotyledonary depression and the embryonal part lying below the uh, cotyledonary attachment depression is the hypocotyl. You can see here. This is the hypocotyl. From this depression between the cotyledonary, the two cotyledon, a plumule will get differentiated and the embryonal part lying below the cotyledonary attachment here is the hypocotyl. Now soon the radicle is also differentiated in the embryo from below the hypocotyl. Okay, you can see here the cotyledons. This below the cotyledon is the hypocotyl, and a radicle is also differentiated in the embryo from below the hypocotyl. Now this embryo, it remains enclosed within a hard covering called a Kolya Raisa. Now a Kolya Raisa is like a sheath, okay? It's like a sheath which protect the root of a germinating embryo. So next here we will we will take a look at the structure of the seed. Structure of the seed. So after fertilization, the ovule will be transformed into a seed. Now the nucellus as well as the inner layer of the integument, they are used up as nourishments for the developing embryo. The major seed appears as orange-red or reddish-brown structure, and it comprises of the following structures. Okay, the seed, it usually appears as orange, red or reddish-brown structure, and it comprises of the following structure. We can see here the first structure, that is the testa. The testa is nothing but it is the seed coat, okay, which will coat the seed. It is formed by the outer brightly colored fleshy layer and the middle layer of the integument. We know that the integument, it has three layers. Remember, the inner fleshy layer, the middle stony layer, and the outer fleshy layer. So the testa or the seed coat, seed covering, it is formed by the fleshy layer as well as the middle layer of the integument, okay, of this integument. So next, what do we observe in a seed is the micropyle. Okay, micropyle, here you can see that in the embryo we have the micropyle. This micropyle, it is present in the form of a small opening just at the top of the seed. Okay, here at the top of the seed there is a small opening termed as the micropyle. Next we have the endosperm. So inner to the seed cone lie a wall tissue called the endosperm. Here, just inner to the seed cone, these layers, they form the seed coat or the testa. Then we have the small opening called the micropyle. Then we have the endosperm. You can see here this part, this whole thing, this part here is the endosperm. So the endosperm, inner to the seed coat lie a wall tissue called the endosperm. Now these cells, the endosperm cell, they store a large amount of food material for the developing seed. And last, what we have here in the middle, last, inner to the endosperm, we have the embryo. So embedded in the endosperm, we have the embryo. It consists of two cotyledons, a plumule, a radicle. Okay, a plumule which will form in the depression between the two cotyledons, and then we have a radicle. Okay, and this embryo will remain suspended into endosperm by a long spirally coiled suspensor. You can see here, this is a long spirally coiled suspender. So the embryo will remain embedded inside the endosperm. Now, thus a major seed of Cycas, it represents three generations. The testa or the seed coat, which is formed by the integument, it represents the parent. Okay, that is the sporophytic generation, diploid, and the endosperm, which represent the second generation, okay, that is the gametophytic generation. Now the gametophytic generation, which is haploid, and the embryo here after the fertilization of the male and the female gametes, which are haploid, after they fuses together to form the zygote, the embryo represents the third generation, that is the next sporophytic generation. So this is from the parent, which is diploid, the sporophytic generation. Now the endosperm is haploid, which represents the gametophytic generation, which is haploid, and the embryo is again diploid, which represents the third generation, that is the next sporophytic generation. Now let us take a look at the germination of the seed, how it germinates. Now we know that the seed will remain embedded, the embryo remain embedded inside the endosperm. So the seeds, they remain viable for not more than a few months. Okay, and under suitable conditions, the seed starts to germinate. It absorbs water and the embryo expands. So the expansion of the embryo due to the absorption of water open the hard seed coat. Okay, so after the embryo absorbed water, it will expand, so it will break open the this hard seed coat or the testa. Okay, so here the Kolya Raisa, that is the seed coat, it protrudes out and is pierced by a growing radical. Okay, a growing radical. Next, what do we have here? This radical, it will grow down and form a taproot or the primary root. Okay, this radical here, it starts to form, and the Kolya Raisa, it protrudes out and is pierced by a growing radical from here. This growing radical, it grows down and it will start to form the taproot or the primary root. Now the cotyledons, they do not come out of the seed coat, okay, but they absorb food from the endosperm for the growing embryo. So here in this case, inside what happened, this radical, it will come out of the seed coat of the Kolya Raisa and it will start to form the primary root, but the cotyledons, they will remain here inside, okay, inside, and they will absorb food from the endosperm cells, because we know that the endosperm cells, they store a large amount of nutrients for the growing embryo. So these will help in the absorption of food from the endosperm. And also here, the stalk of the cotyledons, okay, the stalk of these two cotyledons, they will elongate to carry the plumule, because we know that in the depression a plumule will form. Now the stalk of the cotyledon, they will elongate and they will carry the plumule out of the seed coat. You can see here a small plumule, okay. So these two cotyledons, after they elongate, they carry the plumule out of the seed coat. Now the growing plumule, this small growing plumule here, you can see here in this next picture, where the plumule, At first it will form only two scaly leaves. Okay, at first it will form only two scaly leaves, then later on it will form the foliage leaves. Now if you see here in the developing seed, in the germinating seed, where the foliage leaves, in these foliage leaves we can see that they are circinately coiled. So the foliage leaves show circinate vernation. In both sides of the rachis, the seeds, the leaves of the foliage leaves, the leaves are coiled circinately. Okay, and the seed germination here is Hypogear. Because why? The cotyledons, they remain on the ground. Okay, the cotyledons, they do not come out of the seed coat or the testa. Remember, I told you that the cotyledons, they will remain embedded inside the endosperm. And why? Because they will help in the absorption of food for the developing seed or for the developing embryo. So this type of germination is, we call it a hypogear. Okay, that is hypogear, meaning on the ground. They remain on the ground. Because the cotyledons, they remain on the ground, enclosed in a female protellus or in a female endosperm. So that is why we call the seed germination is underground. That is hypogear. So next, what we will, okay, that is all about the seed germination. Now we know that these are the foliage leaf starts to form. This is here, the first form leaf is actually the scaly leaves, and then we have the rachis. This is the seed, and this is the stalk of the cotyledons. You can see here this one, this actually they are the stalk of those two cotyledons. Actually the cotyledons, they remain embedded inside. Only the stalk of the cotyledon will come out of the endosperm to push the plumule out, so that a leaf will start to develop, to form. So that is all about the germination of the seed. Now in this picture, I just want to discuss a little bit on the life cycle. Okay, we had already studied in my previous videos. I had explained about the all these, okay, the megasporophyll, the microsporophyll, the formation of the megaspore and the microspore, and how fertilization occur, which leads to the development of the zygote, the embryo, and the seed will germinate, which will later on develop into the male and the female gametophyte. So if we take a look at the overall life cycle of Cycas, it actually has two phases. Okay, that is, we call here a sporophytic generation, and okay, one more thing here, a gametophytic generation. We have two generation. How? Because here if you take a look at the zygote, so first we have a male and a female gametophyte. We know that from a female gametophyte we'll get the megasporophyll, and these megasporophyll, we will get the megasporangium, which will contain the megaspore. Okay, the development of the megasporangium and the megaspores. And from the male we have the microsporophylls, the microspore mother cells, and the microspores. Okay, these together, when after pollination, when fertilization occur, okay, here this is during fertilization, how the after pollination, the male gamete, when they reach the female gametes, they will fertilize to form a zygote. So here if you take a look at the male and the female plant, we know that they are diploid till this part, that is the megaspore mother cell is formed, which is diploid, and the microspore mother cells, which is diploid. These will give rise to the male and the female gametes, which are haploid, okay, due to meiosis, okay. The meiotic division, we also call it a reduction division. Why? Because from a diploid microspore mother cell, diploid mother cells, we will get a haploid spores, okay. The megaspores, that is the female gamete, and the microspores, that is the male gamete, which are haploid. So from here starts the gametophytic generation, that is haploid, N haploid. So these, after they fertilize, actually after till here, both the male and the female gamete, they are haploid, okay. So this phase is the gametophytic generation. Now when they fuse after fertilization occur, they will form a zygote. This zygote is diploid, which represents the first phase in sporophytic generation, that is 2N. So a zygote will form the embryo, and the embryo will develop into a seed, which will germinate and develop into male and female gametophyte, which are the sporophytic plants, okay. So if you take a look at the life of a cycas, life cycle of cycas, it shows a distinct alteration of diploid, that is sporophytic generation, and haploid, that is gametophytic generations. Now the plant body, they represent the sporophytic generation, while the gametophytes, both the male and the female, they are very much reduced, okay. The male gametophyte is represented by a pollen tube with persistent prothelial cell and the sperms, and the female gametophyte here, the female gametophyte is represented by an endosperm tissue within the megaspore having the archegonia, okay, that is the female sex organ. After fertilization of the egg by a sperm, we have the oospore, that is the zygote results, which is the first step in the diplophytic phase, okay, that is the sporophytic phase, okay. So this is a diagrammatic representation of the life cycle of cycas. We can see here the graphic life cycle also is the same, where we have the sporophytic generation and the gametophytic generation. So here next I just want to mention a few economic importance of cycas, okay. Few economic importance of cycas. I will just mention some. So here we see that the first economic importance